So good afternoon, everyone. It's a great opportunity to be here, and I thank Lee, Ben, and Kai for giving me this opportunity. And today, I'll talk about how do you integrate genomic selection in a real wheat breeding program, what could be some of the challenges, and what are the opportunities, and how you can transform some of these challenges into steps for the future, or research for the future. So this is a very high collaborative work, and there are several people involved in collecting the phenotyping data, genotypic data, and so let's move. So since this conference is about delivering genetic gain in wheat, here is a brief overview of how we could deliver genetic gain in wheat using genomic selection. Dr. Ben Hayes has given an excellent talk about this morning, and I just wanted to show you one quick slide of how genomic selection can be used to deliver genetic gain to farmers. So initially, you have a population, which is known as a training population, where you have lines that have both genotypes and phenotypes. And then you train your prediction models, and you obtain the GEBVs, or the genomic estimated breeding values of new lines or your new germplasm. And you can call this as your breeding population. And then you select lines with the highest GEBVs. You make crosses, advanced generations, and this kind of will help you reduce the cycle time by rapid cycling, and you can rapidly deliver varieties to farmers. You can also take a simultaneous path here. Once you train your prediction models, you can also evaluate the performance of your prediction models, and I would call this as a pre-implementation phase, where first you need to test your models, and I would call that as a validation population, and then you can really start implementing genomic selection in your breeding program. So to implement genomic selection, I would like to ask all of you, so assume that you're breeders and you would like to implement genomic selection in the breeding program. So what are some of the first steps that you would take? What would you like to do first? What would you like to know about genomic selection to implement it?
Trevor told us to set up a database.
Okay, great, yeah, sure, you should do that, yeah. And readers here, what else would you like to do to see if this technology is helping you with your breeding program?
Developing own training population.
Okay, developing your training populations, great. Any other ideas you have? All right, great, yeah, those were great points, and here I've listed some more things that I would do. So when I came to this job a year ago, so all this is going to be a year of my GS work at Summit. So my boss put me as a wheat breeder, so he gave me that position. But it's kind of really very helpful to me because now I can think of genomic selection from a breeder's perspective and look at what might help a breeder, what might not help, and how we can improve the things that might not help. So these are the things that I would do. First, you identify opportunities for implementing GS in the breeding program. It can be different traits, different diseases, or quality, or yield. And then you evaluate genomic predictions for these traits and different stages of your breeding program, for different populations that you want to evaluate. And then you find the relative advantage of GS over your current strategies of breeding, your phenotypic selections, or any other strategy that you might have. And then implement GS for traits where your relative gain will be higher. And if you have challenges, this is the most exciting part, and I'm currently in this phase. There will be challenges to implement a new technology or with any research. And what you do is transform these into research opportunities. So I'm currently looking at a lot of challenges that we have and how we make progress despite these challenges. And first, let's start with identifying opportunities for implementing genomic selection. So here I'm going to give you the example of the Summit wheat breeding program, but many of your breeding programs might be similar, but maybe of a smaller scale than we do. So you can have parallel comparisons to your breeding programs as well. So the first stage, when you select parents, GS is a good starter here. You can select parents based on those that are known to have high GEBVs and use them in crossing. And then the next stage are your early generations. So you have generations from F1 to maybe F5, F6, F7s. And one of our challenges with Summit is that our breeding program has quite a lot of plants. You have millions and billions of plants. So when you take the cost of genotyping to be $20 per sample, it's going to cost us about $20 million. And as Susanne mentioned in the morning, sometimes the cost of line development might be about $5 that Breeder told us. So it's hard to kind of justify that with $20. And so genotyping has to still become cheaper given a large-scale breeding program. And again, there might be some logistical constraints because currently we don't do internal genotyping. We outsource it. Jesse Poland does it for us. And there might be also high heterozygosity in these generations, and plants will segregate, of course. So you have to be careful considering these stages, and we will look to do something with these stages if you can use lower density markers, or if you could take subsets of populations from these stages and test genomic selection, but we haven't done anything so far in these stages. And then moving on, we have the next stage, which is before yield trials, before your testing, yield testing stages, and that's the pre-yield testing. And again, you have about 70,000 head rows there. So this is again a huge number of lines for genotyping, and they are usually grown as small plots, 1 meter plots, and then in the yield trials, you'll have larger plots. So one thing we've done here is we are testing pedigree-based predictions and combining that with high throughput phenotyping. So using both these strategies to see if we could predict how well these lines will yield in the next year. So for some of these lines, we do have high throughput phenotyping data and the yield data in the next year. So we do an experiment to see how the high throughput data in this year will translate into yield the next year. So that's one of the objectives of the DGGW project here, and we're testing that. And the next stage is the yield trial stage, the first year yield trials. So we have about 9,000 lines in this stage. So as part of the USAID project, we've genotyped all these 9,000 lines for the past 4 years. And this is done in collaboration with Jesse Poland from Kansas State. And we do have data for grain yield, agronomic traits, and stem rust in this stage. So we have currently tested GS at the yield trial stage. And then from the yield trials, you go on to the elite yield trials. So 10,000 lines, and then you go down to 1,092 lines, which go into these EYTs. And you do a lot of phenotyping at this stage at CIMMYT. So you have grain yield, 3 reps, 6 different environments, 2 optimum environments, drought-stressed environments, heat-stressed environments. And then you have agronomic traits, and then you have different diseases which are phenotyped, and also quality is phenotyped at this stage. So you have a lot of data on this stage, and here, these 2 stages are the ones that we are currently testing genomic selection on. And first, let's move on to some of the results we have. So looking at the stage for the yield trials, where we have 9,000 lines and each of the yield trials, and as Dr. Ben Hayes mentioned about cross-validation accuracies, so in a 5-fold cross-validation, just say you have 10,000 lines, you divide it into 5 folds, and you use 4 folds to predict the 5th fold. So you're basically using 8,000 lines to predict 2,000 lines. So these are the accuracies, and the numbers here just denote the cycle. So YT 1314 denotes 2013 to 14, and so on and so forth, the different years for the past 4 years of our yield trials. And the accuracies for grain yield look very good. However, our breeder was interested in knowing what happens if you predict different years using another year, because that's what a breeder is interested in. What can you do if you don't phenotype these lines? So that's what the accuracies that came from predicting the year 15-16 from the year 16-17, or vice versa. So this will be a backward prediction, and this will be a forward prediction. And our accuracies were about 0.3. And so that's the example for grain yield. So what results in these low accuracies across years is one of the questions that we wanted to look at. So that's again transforming this challenge. So we don't have an accuracy that is high enough for us to really implement genomic selection at this stage. So what was going on here? So I did a quick GWAS at this stage and looked at all the 4 different nurseries, and you have to take this into account. So this is about 10,000 lines in 4 different years, and this is grain yield. And you can see how complex of a trait it is. We did not have a higher, if you look at the minus log 10 p-values, they're pretty low. and there are loci all over, which shows that it is a complex trait. And some of our lines are well fixed for the grain yield loci because you use elite parents. So in such a study, there might be difficulty in getting good loci for grain yield. So this might be one of the reasons why our accuracies across years are not very high, because you might have different loci affecting grain yield, and that might be primarily because of using different parents. Because the last 2 years, Borlaug was one of our key parents in crossing, but previously you had different parents, so that might be different. So allele frequencies change in these different nurseries, which might result in a reduction in accuracies or decrease in accuracies. However, we did this for stem rust as well, and this is UG99 stem rust, phenotyped in Kenya, and Sridhar and Dr. Julio is here, who are all involved in phenotyping this. So we did this for stem rust, and our cross-validation accuracies looked good. They were about, the highest accuracy we could get was about 0.66. This is kind of similar to what Dr. Hayes showed in the morning. And across nursery accuracies, and this is again, you have to keep in mind that these are 10,000s of lines, and they looked pretty good. This looks very promising, so you can be 50% sure about some of your lines in these yield trial nurseries if you do genomic selection, which was very promising for us. So we did this, and the next thing that we also tested was genomic selection in the second year yield testing stage. That's what we called as a elite yield testing stage. And here we have 6 different environments at Summit. So this is a bed 5 irrigation, which is optimum irrigation, and this is flat 5 irrigation, which is the same but in a flat planting system. And you have 2 irrigations, drip irrigation, early heat, and late heat. So these are the 6 different environments that Summit evaluates these lines for. And as you can see here, the within nursery cross-validation accuracies are always high. But when you start predicting across nurseries, these are like the highest accuracies, like the best accuracies that you could get using different data sets, and those are pretty low. That's what we currently observed. And then I wanted to look at what is the effect of using these different training populations. So here I have 3 different training populations. So if you use lines from the same nursery, which is nothing but cross-validation, and then if you use lines from the previous nursery, which are those in yellow, and if you use lines from all the previous nurseries, so that is using 3 or 4 years of previous year's data. And when we did this study, as you can clearly see, using lines from the same nursery always resulted in the highest prediction accuracies. And when you go a year back, or even if you increase the size of your training population, for a trait like grain yield, it might be challenging because sometimes it could be just more noise into your training sets because the lines those years were completely different from the year which you're using to predict that data. And so one thing I looked at, so what is causing these poor predictions across years? And when you look at this figure, so these are 1,000 lines that are common in the first year and second year yield trials. And these 4 different panels here show different nurseries. And as you can see, the phenotypic correlations, these are just phenotypic correlations. And if you look at them across years, they are very low. So they can range from 0.2, 0.3. And how many of you breeders think this is not a reality? Have you seen a line that is yielding very high the first year and it does not yield well the next year? So that's pretty much a reality. And that's again a problem. And when you look at a trait with low heritabilities, we should always keep in mind that the heritable part of the genetic variation is what can be predicted as well. So you have, let's say your heritability is 0.2 and 80% of the variation is due to environmental effects, then you might not be able to predict some of those effects. For sure, there are G/E models, but I can show you what problems we have given the models that we are dealing with. And this is correlation between grain yield in different environments. And these are the 6 different environments that I've mentioned previously: drought stress, heat stress environments. And you can see the correlation between the lines evaluated, the same set of lines evaluated in these different environments can be quite different again across the years. And one more, so again, the question is, what is leading to all these differences? So it could be G by E, and I would like to categorize that into G by L, which is genotyped by location interactions, and G by Y, which is genotyped by year interactions. And we do not have very much of a location interaction because all of these lines are evaluated in Ciudad Obregón. So the location is kind of fixed for us across the years, but there is a huge impact of G by Y interactions. And if you look at some of the 10-day temperatures during the growing season, one of the seasons might have a very warm temperatures during the growing season and then gradually fall towards the end. And another year might have quite high temperatures during the critical grain filling phases, and then it might be very variable. And if you have, since we have readers here, this is something that we commonly observe. You can have very warm temperatures in the beginning, and then this is not exactly what we see in Obregón, but sometimes in some parts of the world, this is a reality. And then the temperature changes, and plants have different responses to changes in temperature. And you might not be able to capture all that variation. So here's what I concluded here. So grain yield is going to be complex. It is complex and it is challenging, but this applies to both phenotypic selections and also genomic selection. And if you have a lack of correlation among the years, then it's certainly going to affect our ability to predict these lines. And accurate estimations of G by Y interactions will require nurseries being replicated in different years so that you can get a sense of how much is the G by E interaction, which sometimes you do not have in a breeding program. And that's something that we really need to focus to train our models with good replicated data. And so here's again another question. So how easy it is to predict the weather? So how many of you have been fooled by a weather app? Like you take an umbrella to your office and it doesn't rain, and you forget taking an umbrella because it said 100% sunshine, and then it rains. Has everybody gone through this, or some of you, depending on where you're coming from? So that happens always, and sometimes it's hard to predict the weather. What kind of weather do you get during your crop season is kind of very hard to predict. And this is a challenge to G by E. It's a real factor that has to be considered when we make predictions for a complex trait. And how do you deal with this challenge? So we're not here to talk about these are the challenges, but how do you deal with this wisely? So the problem here is that we have models that are trained with the data that's quite variable. So grain yield is different in the next year. And we train a model and we predict it and compare it with another set of data that is also variable. So when you do this, your accuracies are naturally going to be low. One way to deal with this is we increase the reliability of predictions by using well-replicated data, and you should have a large enough training population with an ample diversity as well. So what we understood is that we need good replicated data for a trait like grain yield, which is so variable across years. And again, when you estimate the accuracy from single year performance, that's going to be biased because that single year's performance of a genotype is not going to be its true breeding value. It can be where if it's going to change the next year, it's important that you either correlate it with multi-year performance of a line or simply use good training populations and take it by faith and move on with your predictions. So yeah, that's what we currently observed. And then now moving on to diseases. So a lot of you work with stem rust here, and these are our stem rust prediction accuracies. And here the first, the tallest bar in every, the four tall bars will be cross-validation accuracies, and the other bars are prediction using different populations. So as you can see here, some of the populations are very well predicted from the other nurseries. So stem rust is not such a complex trait, at least in our populations, as is not very complex like grain yield. And if you can look at one of the nurseries, which was predicted from another nursery, the accuracies were about 0.6. And this really looks promising to help us reduce some of the replications that we have across different seasons. You can take GEBVs instead and move ahead with your selections. So genomic selection is not all about prediction accuracies. So I wanted to move further after you get your accuracies, so how effectively you can use your GEBVs for selections. And this is what I did. So we compared GS with phenotypic selections, and these are just cross-validations within years. And I've categorized them by lines that were not selected by GS and phenotypic selections because they are poor yielders, and then the lines that are selected by both these methodologies, and there will be some that are just selected by phenotypic selections but not by GS, and then there will be lines that are just selected by genomic selection and not phenotypic selection. And if you look at this graph, pretty much about 68% of the lines, a maximum of 68% of the lines will be selected by both these techniques. So the reason I did this is for a trait like grain yield, where your variation can be very narrow. You can have 5 tons to 8 tons in your advanced lines, and you cannot expect every time to get a 5.6 and a predicted value to be a 5.6. You need to look at how effectively you can use the breeding values that you have. So that's why you need to look at what are the possibilities of selecting from the GEBVs that we have. So, and it looked pretty promising. Even at an accuracy of 0.51, you can select 68% of the lines and also discard 68% of the lines. But here, I'm using a selection intensity of 0.5. So why did I use this intensity? So here is the reason. The next slide. So when you use a selection intensity of 0.1, you discard 90% of the lines and you select 10% of the lines. So when you do that, you kind of get an inflated estimate of the number of the percent of the lines that are going to be discarded, because you're anyways discarding 90% of the lines. So you're going to get a high percent of the lines that are discarded. And if you look at a selection intensity of 0.1, so this is a percent lines discarded, and this is a percent top lines selected, and this is for one nursery, and this is for the other nursery, and both these are just within populations or based on cross-validations. So you will be able to select the top 30% of the lines, and you will be able to discard 90% of the lines at a selection intensity of 0.1. And if you want to do a more fair comparison, then you can use, I've just used, again, a selection intensity of 0.5, where you can select an equal number of lines and discard an equal number of lines. So you can very well compare how many lines you can select or discard. And when you look across nurseries, that's where the percent of top lines selected by phenotypic selection and genomic selection kind of decreases because we have low accuracies across years. But one nice thing to note is that even though your accuracies can be low, you still can select and discard a reasonable number of lines using GS when you compare it with the phenotypic selection. But there is a caution here. All these selections are assuming that the phenotype is a measure of the true breeding value. And this, in the case of a trait that is highly variable, might not be true because you really don't know what the true breeding value is. P is equal to G plus E, and this could be, a lot of this could be due to the environment. So that's what you need to keep in mind when you look at comparisons between phenotypic and genomic selection. And I'm again showing you for STEM-RUST that I've showed you before. It's going to be, these are some of the promising results that we have for STEM-RUST. So at a accuracy of about 0.6, you are able to discard 94% of the poor lines, of the susceptible lines, and select about 44% of the top lines, which I think is really good. So you have an accuracy of 0.6, I would still say you can go ahead and do genomic selection for traits like STEM-RUST. And again, this is what we see at a selection intensity of 0.1. This red region is a number of lines you will discard by genomic selection and phenotypic selections as well. And the green area is what you would select by these by both these methods. And that's what we see. And I have some questions here. So how well do you think we can predict the top performers in your breeding population? Does any breeder or anybody has any opinions? How well can you do this? Because there's always my observation was that there's always going to be this area here, a few lines which are very performing very well, and you might not see it in predictions. And why do you think it might happen? Does anybody want to comment on? Okay, yeah, maybe I'll just... So I like this cartoon because sometimes it explains what might be the problem. So what we predict is always based on known information. But sometimes you will have a few transgressive segregants, and you might not be able to pick those up just because your models are trained with known information. And you will always have some lines like that. And I would say you have to expect it in predictions. And so currently what the breeder does is he takes this portion, which is lines that are selected by genomic selection as well, that are known to have high breeding values, but the phenotype was not that great. So currently the breeder gives it another chance. So he includes them in the next year yield trials and reevaluates that performance to see if that goes well. And so the next thing we did, yes?
It's a bit of a conundrum because you just said that the phenotypes are not the real breeding values.
Yeah.
Which a little bit of me just died on the inside when you said that.
Yeah. Especially in a large breeding program, that is something you might expect. And for a trait like this, you might expect. Yeah. Yeah, so the next thing is to evaluate genomic selection with our current strategies. So CIMMYT has a very good pedigree system. So we maintain pedigree records of all the lines in our nurseries. And so we compared genomic selection with the pedigree-based system. And I'm showing you two different traits here, one being grain yield, and then the next is stem rust. And this is a cross-validation accuracy, which says AYT 15, 16. And this again is a cross-validation. These are cross-validation accuracies, but the rest are going to be predicting different years using different populations in other nurseries. And the green ones are the pedigree. So what we observed is that the pedigree does equally good as the markers, both within and across nurseries, which is not something that you would expect because the markers are known to capture the realized relationships between lines, and they should perform better than the pedigree. So if you take a look at the genomic and pedigree relationship matrices, so the top panel, so these are scales between 0 to 1 just for comparisons, and the top panel is the genomic relationship matrices for 3 different populations, and the bottom panel you can see the pedigree-based relationships. And a dark blue here would mean that there are no relationships, and if it goes towards the greener side or the yellow side, it's going to show that there are good relationships. And these are hot spots that you would observe if you have highly related full sibs, or if you have a number of full sibs in that nursery. So that's what we observed. So that clearly tells us that the pedigree is having a 0 relationship for unrelated lines, or a 0.5 for full sibs, as Ben mentioned in the morning, or a 0.25, it's going to be a half sib. And the genomic matrix is able to capture that well because it's capturing that variation based on IBS, or identity by state relationships. So that's okay. So that's what it has to do. It's doing that well. And we did this analysis comparing what happens when you don't have any full sibs in the training population, and if you just have one individual cross, and what happens if you make predictions with 2 or more full sibs, so that you have one full sib in the training population and use that to predict all the other full sibs in that population. So we have compared the genomic and the pedigree-based relationship for these 2 different scenarios. And here are the different nurseries. And as you can see, just a general comparison between this figure and this figure, you can see the big boost that you get if you include full SIBs in the training population. It's kind of really telling you that you need to have some amount of genetic relatedness in your training sets to be able to get good prediction accuracies. And here, there are some clear cases where the pedigree is not able to predict well and the genomic relationships are very good. But what came as a disappointment is this figure. So if you have full SIBs, then there is a certain amount of Mendelian sampling variance that your genomic relationship matrices will capture, but the pedigree will not capture. So that is what we expected. But the results turned out to be slightly different. The pedigree seemed to be as good as the markers in even capturing that within family variation. So we have a problem here, and we wanted to investigate more on why this happens. And it turns out to be something that you would expect, but you might not have really expected. It's kind of something. So we looked at the family sizes and the number of families in these 4 different nurseries. And it seemed that about 50% of the lines in each nursery had no full SIBs. So using 50% of the lines which have just one individual per cross, and the remaining 50% that had at least one full SIB, you had lines that had minimum variance for grain yield. And this was because the breeder had selected them. And this tells us very clearly that the stage where you test genomic selection is also important. For just because they were selected, and this is something that is very obvious, but anyways, since this is a stage where we had well-replicated data, we had to use this stage as well. But since these lines were highly selected, the SIBs were all similar. So the pedigree is doing as well as your markers would do. And this is something that is very important to consider. So GS will be less advantageous in a stage where you have minimum Mendelian sampling variance among your full SIBs, as you simply don't have any scope to outbeat or outperform the pedigree-based selections. So this is kind of a paradox here. So the most affordable and the well-replicated stages for training models had little advantage over the pedigree because of the family structures in these nurseries and because they were highly selected lines. So you have to be very careful at the stage where you implement genomic selection in your breeding program. And there are some stages, like for leaf rust, if you go down to these generations, I would not do genomic selection there because these are highly selected traits right from the beginning. So you might have very little variability. So the stage you test it is very important. And we wanted to test this more. So one other reason why this could happen is that your markers are not capturing the relationships well. So that could be, again, another reason. So we wanted to check the effect of these markers. So all this data is based on about 2,000 markers. So we did this experiment where we looked at markers, the whole set of markers, 2,000 markers, and then we grouped markers, just removed one random marker if they are highly correlated. So if you have R-squared values of 0.8, 0.6, 0.4, and 0.2, and if you look at these results for grain yield, and both within nursery predictions and across nursery predictions, one thing that you can observe is it only drops down if you come down to a 29 markers, which you would expect. But even if you use 250 markers that are an R-squared of 0.4, you still are able to pretty much get the same prediction accuracies. So what does this imply? So all the information that we have for predictions are just because of about 250 markers. So that could be because of 2 reasons. The high LD in wheat, as Kai was mentioning, or because GBS markers are kind of highly redundant, which anybody working with the markers will know. So these are 2 reasons why something like this could happen. So the next thing we looked at is the distribution of these markers. So these are the different chromosomes. So you have chromosome 1A, 1B, 1D, and this is the physical map of the wheat genome. And this distribution seemed pretty interesting. So most of the markers are concentrated towards the telomeric ends, and this gives the marker density. So if you have a red color here, it would indicate you have more markers there. that region has a higher marker density. And you can see there are vast stretches of DNA where you don't have any marker coverage. Is this because of the nature of the wheat genome? Because there are studies that say most of the genes in wheat are concentrated at the telomeric ends, and there is not much recombination at the centromeric regions. But it could also mean that the markers do not sufficiently cover, and since genomic selection is based on the assumption that you have one marker every centimorgan, but I would also say that we have to do it actually with a genetic map versus a physical map, because in physical map you can highly inflate your distances because you never know if this translates to a centimorgan or more, and it is different in different parts of the wheat genome. So that's again one of the things that we observed, and this was aligned to the RefSeq version 1. So, and then again, so the markers might be some sort of a problem. And again, the next question was, so what is the relatedness across these 4 different populations that we use for predicting? Are these completely different populations? So we just did a principal component analysis, but these are just based on 2,000 markers, and then I would like to mention that. So, but still the markers captured about 8% and 6% of the variance. And what you have in different colors here are the different nurseries, the EYT 13-14, 14-15, and the other years. And that pretty much shows that there's not much of grouping of different nurseries. So then that is what you would expect in a breeding program like CIMMYT, because you have several parents that are recycled in the different crossing blocks. So you can't expect the lines to be entirely different from each other. But this kind of tells me that they're just all over, and I don't see any clear grouping of nurseries. There might be the genetic relatedness is not very different, but once again, this is based on few markers. So, and one other thing that we wanted to study is what are the other reasons for these low prediction accuracies. And one thing we did is look at relationships between grain yield and other agronomic traits. For example, if you are a breeder and you go out of the field, you have a lot of variation in phenology. You have lodging in some lines and you don't have in the others. So a trait like grain yield cannot be considered as a single trait. You always have to take a multi-trait approach for grain yield. And this is a kind of interesting figure. So the different panels here are the 4 different populations that we had. And what's on the x-axis are the days to heading, days to maturity, height, and lodging. And this is just one of the environments in our second-year yield trials. And as you can see, this is again a clear effect of the weather. So one year you have a pretty good correlation with days to heading, and another year you don't have any correlation. And one year the lines are very early, and again in another population you have late lines. And you have 2 years, you have good lodging, you have, and the next 2 years there was no lodging. So a breeder has to take all these traits into consideration when you do genomic selection. And so what we did is, so all this credit goes to Dr. Jose Crasa there and Oswald. So they implemented a new approach here. It is called the item-based collaborative filtering approach. And this is what they use in e-commerce websites like Amazon and Netflix to predict the user's preference for items based on data that they have, the multi-layer data they have. So if you get a recommendation for Amazon for what you're gonna, for purchasing something, it's based on your past searches and things like that. So we wanted to test this model. So if user 1 has a preference for some items and user 2 has preference for other items, and what will be the likely preference of a third user? It's all based on data and similar correlation between items, and which we took it to our advantage in wheat breeding, and it's going to be based on correlations between traits. And this analysis was done by Dr. Crasa's team. So if you just use the genomic relationships, these are the accuracies that we would get. And if we would use genomic relationships in addition to these traits, especially across years, you might get a slightly better accuracies, because you're now considering some other factors that can explain your grain yield variation as well. And so this is just a pilot approach, and our ultimate objective is to use this IBCF with big data, because the IBCF is mostly developed for big data sets that companies or Amazon has. So we have about 70,000 lines that go into our small plots, the pre-yield testing stage. And we have high throughput phenotyping data on all these lines. So as I mentioned previously, one of our objectives at this stage was to incorporate high throughput phenotyping with predictions and see how well you can predict the grain yield. So this is all done by Suchi Smita, who is our physiologist in the breeding team. And then, so our objective was to use some of these different high throughput traits in an IBCF approach and predict grain yield just based on similarity of these traits with grain yield. And what are some of the similarities that you would expect? So here are two different stress environments. One is the drought stress environment, and then you have the heat stress environments. And you have correlations of the high throughput phenotyping traits with grain yield. And as you can see, these are the correlations of the vegetative stage, the phenotypic correlation and the genetic correlations. And you have the correlations at the grain filling stage. And they look pretty good. The correlation of the grain filling stage is about 0.4. And if you look at the heat stress environments, you still have a higher correlation. So we use different models here. So one is the genomic model, and then you have the pedigree relationship-based model, and then you have a combination of genomic and pedigree, and then you just have GNDVI, which we used in the IBCF approach, and then you have genomics and GNDVI, and pedigree and GNDVI. And if you look at all these results, again, what we see is either you use both markers and the pedigree, or we use just the pedigree and GNDVI, which resulted in highest accuracies. But they were more or less similar. But this kind of gives a good indication that we can use this if we have large populations, then we could use pedigree with GNDVI. And I'm just going to tell you, you should take a little caution when you do these predictions. For example, in the drought stress environment, one thing that you would often see is there's a very high confounding effect of phenology and plant height in your stressed environments. So when you do genomic selection, you have to consider these traits as well. So when you do not adjust for covariates, you will get really high accuracies. That might be because of the major genes affecting these traits. And I just looked at some of the GWAS results, and it's just clearly 5B, 2B, the vernalization and photoperiod sensitive genes, which might result in high accuracies. And then when you correct for days to heading, your accuracies might drop down, and it might even be lower than half the accuracies that you get. So when you use populations, breeding populations especially, where your range of phenology can be as much as 20 to 30 days, you need to be careful when you use such kind of data, because days to heading might be a confounding factor. And so one of the questions that I also had is, what kind of prediction models should we use? A lot of people ask. So there are different models, as Dr. Ben Hayes mentioned in the morning. So you have ridge regression, you have Bayes A, B, CPI, Lasso, RKHS. And this is part of my PhD work. And this is just leaf rust seedling, Septoria adult plant resistance, and stripe rust adult plant resistance, and tan spot seedling. And sometimes you might just end up with the same accuracy. It might be different for different traits, but for some of the traits that we are dealing with, the accuracies are going to be more or less similar. So this is just telling you, don't spend a lot of time in comparing the different models or using different models. Just use GBLUP or RKHS or other models that you like, and they're going to be very similar. So, and I also mentioned this because sometimes you get asked, the models that you're using are bad, but it could not be the models, it could be the data that goes into the models that sometimes might affect your predictive abilities. And this is just showing you the marker effects for these different models. Again, as Dr. Hayes mentioned, you have different prior assumptions when you use these different Bayesian models. And this is just showing you Bayes A, B, C, pi, Lasso, and the Bayesian ridge. And this is also part of my PhD work. And this disease on top is Septoria trudesi blotch, which is, and we have then the data is for adult plant resistance, and the bottom panel is for tan spot, and that's going to be seedling resistance. And as you can clearly see, if you have large genes, some of your models based on the assumptions might capture that well. And if you have many genes, small effects, then that's going to give you something like these if you look at the effects. And we also did a comparison of two different marker platforms. So this was a comparison between GBS markers that we get from KSU and .seq markers, which are technically GBS markers, but they use a slightly different restriction enzyme. So our objective was to see how well these two marker systems compare when you do genomic predictions. And as you can see here, the red line shows .seq, and the green one is GBS, and the blue one is using both these markers together. So what if we use 10,000 markers, using from both these marker systems? And one interesting observation is that you might not necessarily increase your prediction accuracies just by increasing the marker number, especially if those systems are going to capture redundant information. So it could be that both these systems were capturing the same information, and that's why you don't see a very big increase in accuracies using both these marker systems. And so moving on to the last section of my presentation. So we do GS for quality traits as well. And this is what really is very interesting, because when you look at these different quality traits, the alveograph, flower protein, flower yield, loaf volume, or mixing time, and you can see the red ones are always within nursery cross-validation accuracies, and the green ones are going to be across nursery accuracies. And as you can see, quality is very well predicted using our marker system. And even across nurseries, you get very good accuracies for quality. So this is really promising, and a PhD student from Jesse's lab, Emily, is working on these quality predictions. And so I also want to look at how complex these traits are. So if you look at the alveographs, they are kind of mostly governed by the glutenin genes on 1B or 1D. And if you look at flower protein or flower yield, or even loaf volume, they are pretty complex traits. But one thing you have here is you don't have a very high G by E interaction. So the accuracies, even for complex traits, are still going to be high. And we also compared genomic and pedigree-based predictions for quality. And the red line here is pedigree, and the green line here are the markers. And that's quite promising. The markers seem to, the genomic relationships seem to be very good if you're using GS for quality. And although quality is not a very primary target of our breeding program, I'm still sure that there are many companies or breeding programs that primarily focus on quality. And GS can be very good if you use it for quality traits. And moving on to this section of implementing GS. So I've showed you so far the results we have for Simmons breeding program. And what do you do? So this is moving ahead to, all this was like GS 1.0 and moving ahead to GS 2.0. So one thing we could do is utilize all the data we have. Now we have genotyping data for about 40,000 lines. So what you do is utilize all this data, and then there are also markers that we have for stem rust and other traits from our big data set GWAS. And a key thing that we are interested is doing genomic selection right from the parents. So we've all been testing it in stages where we might not have a very good advantage. So this is what we are looking forward to move. Genotype, so we are currently genotyping our parents, and Susanna is doing that for us. So once we have the parents genotyped, we are going to use some of these data sets and try to predict the parents and their progenies, and kind of include the parent information for lines where you don't have full SIBs, and see how that can increase your predictions, and design some of objectives in these stages. So at this stage, we are still not yet there, where we don't have a very good or cheap genotyping technology to be able to do within family selections, but we are really interested in studying this. What is the role of within family variance? Because F2s is a stage where you have the maximum within family variance. So what is the role of that variance? What is the role of across family variances here? Can you predict across families here, just using some of the parental information? These are all the different things that we're looking forward to test. And as I mentioned, dealing with this big data. That's what Dr. Kraza will be doing. So use some of this item-based collaborating filtering technique and predict the lines using HTTP data from drones that we are collecting now. And what do you do? One other thing that you could do, although we do not have good accuracies for grain yield, you can still scale up selections at this stage. So typically our quality tests are all done in these 1,000 lines, but you can scale up selections with GWAS. So these 10,000 lines you do not have any quality data for. And our quality specialist, Carlos, told us it's going to cost about $61 for a sample of quality evaluations. So in stages where you don't evaluate quality, you can easily bring genomic selection there. So that's going to be a good cost saving for the breeding program. And then you can use it as an additional replication for stem rust. So you just do one replication of stem rust, and it's going to cost you $15 per sample. And if you just use the GEBVs, you still will gain a lot by capitalizing on the genetic component of that variance that you see. So you can substitute replications within years because the breeder still wants to have at least one set of observed data, but you can simply substitute the other replications with genomic selection. And then you use GS for multiple traits, because if you have 10 different traits and $20 per sample, it's going to cost you $1 per sample. And you can easily justify that with what you do. For example, a replication for grain yield is going to cost you $10 per unit of evaluation. So if you have 1,000 lines, $10, 6 environments, that's again going to be $65,000 of savings. And you can easily do that with genomic selection. And again, with stem rust, we currently do about 4 seasons of evaluations, and we can use GEBVs to select for stem rust as well. And moving on to this last point, and this is what exactly the breeder brought up in the morning. So this is a correlation between the different traits. So 1,000 lines, and a breeder has to look at all these different traits when he makes selections. And the color code here is, it's going to be negative, highly negative if it's going to be brown. It's going to be close to 0 if it's going to be yellow or the greenish area. And if it's blue, it's going to be a very high correlation. So a breeder looks at multiple traits. And as you can see here, some of these traits, like disease resistance, might have negative correlations. If you take a biotrophic disease and a disease caused by a necrotroph, usually like tan spot and rust, it might have a negative correlation. And this is very important when we do genomic selection. So genomic selection for one trait might not help us. And so we're really looking forward to have good training sets for different traits and use something like a selection index and be able to predict all these traits. And that brings us to my point that this is probably my last slide, and we need a broader vision for GS. So instead of focusing on one single thing, sometimes in a large-scale breeding program, you will need to look at several different things. And we should be able to understand if it's fit in the breeding program. And so these are all the pyramids in Mexico, if you've been there. And another thing I would say, so when I first climbed these pyramids, it looks very steep, and you will have some fear. Are we able to go into? Are we really able to make it to the top? But the characteristic of any researcher or a wheat breeder is that you have to face all these challenges undoubtedly, and you just be passionate to move forward. There are challenges, and I do accept it. But research, that's why we're all here to do good research. So we will be able to get to the top if we persevere with the research that we do. And this is my last slide. And I thank the Bill and Melinda Gates Foundation. So I'm actually funded by the DGGW project, and I work for CIMMYT. So USAID, DFID, they're all very big contributors to this project. And Dr. Ravi, who is our main breeder, who is an excellent breeder, and Dr. Kraza, who is here, who is a very, very great support to me in all statistical models. And we have Dr. Julio there, who is a very good expert. And I have several questions, and he always has answers. And Sue, who does a lot of phenotyping, and Sridhar, who does stem rust phenotyping, and Susanne is here, who does most of the DNA extraction, and then Dr. Pavan, Carlos, Velu, and Leo. And then we also have the team from Cornell, Ronnie, Mark, and the DGW team, and Maricelis, and then Jesse Poland from Kansas, and we have Paulino and Oswald, who are other statisticians who are involved in this project. And this is our last slide, and we are gearing up towards excellence in genomic selection research. And at Summit, since we translate research into impact, we are very much interested in making the best use of this technology, which is very beneficial to the breeder, not to disrupt what the breeder is doing, but to identify areas that it can really help the breeder in doing faster selections. And we are also looking forward to integrate speed breeding, and as Susanne is already doing, and combine GS with speed breeding and other technologies to see and run parallel cycles and see how fast we can get to what we're looking at. Thank you very much.